Lecture 6, Part 3. So, enzymes. How do all these chemical processes help get pushed along into happening? It's through the use of enzymes. So all these reactions that we're going to talk about in the following lectures, cellular respiration, fermentation, photosynthesis, these all happen again through not just one reaction, it's actually a series of reactions, and that is helped along by enzymes. So spontaneous reactions, remember, they just don't need added energy, but they can be slow. They aren't always fast and immediate. It's like we kind of think of the word spontaneous. Remember that when it comes to energy and stuff like that, spontaneous reactions just don't need added energy. But they can still be slow, really, really slow. They will hardly happen on their own in a short amount of time. So for example, sucrose is a sugar, and through hydrolysis of sugar, you break sucrose into glucose and fructose. And so that's technically spontaneous. It doesn't need added energy to happen. But at room temperature, it would just sit for years without appreciable hydrolysis. So again, spontaneous just means no added energy, not that it's going to be fast or anything. So in this example of sucrose hydrolysis, it could happen on its own without any energy added, but it would take a long time. So enzymes help these kinds of processes happen a lot more quickly. So enzymes are or serve as catalysts. A catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. So you can kind of speed up a reaction by adding a lot more sucrose, but that's not going to make it that much faster, and then having that sucrose, it's going to get used up in the reaction. So catalyst is something that speeds up a reaction, but doesn't get consumed by the reaction. And so an enzyme is a macromolecule, usually a protein, that acts as a catalyst to speed up a specific reaction. So in the example again of sucrose hydrolysis, there's an enzyme called sucrase that breaks down sucrose into glucose and fructose and helps it happen within seconds. So it really helps reactions happen faster. And so every chemical reaction involves breaking bonds and making bonds. And so sometimes it would take forever for that to happen on its own. Not forever, but a long time. So a molecule must be, in a lot of cases, twisted into a highly unstable state before those bonds can break and start the reaction. So to reach the state, that molecule must absorb energy from its surroundings, and then that energy might help twist it into an unstable state and then break the bonds, and then the reaction can happen. And so the initial energy, input that is needed to break the bonds of reactants is called the activation energy. It's kind of what activates this reaction. And so heat in the form of thermal energy is usually what's absorbed from the surroundings and often supplies that activation energy. And so molecules become unstable when enough energy is absorbed to break those bonds. And so this is the transition state, when it's unstable enough to start breaking the bonds. And then as atoms settle into the new, more stable bonds, energy is released to the surroundings. So in an exergonic chemical reaction, the formation of new bonds releases more energy than was invested in breaking the old bonds. So that's why there's a net release. So in this example, here's molecule AB, and here's molecule CD. And so these are the reactants in this chemical reaction. Some energy has to be put in and it's usually, again, in the form of heat. And so these bonds are like unstable, and so this is the transition state. And then this is what pushes those bonds to break. And then now A and C can form, and then B and D can form. And so that is, the activation energy that has to be put in for this reaction to happen. And then it ends up overall releasing energy. So that activation energy is kind of like a barrier that determines the rate of spontaneous reactions. So for some reactions, the activation energy, again, EA, activation energy, is low enough that thermal energy at room temperature is enough to overcome the activation energy barrier, but that's some reactions. Most reactions actually have a high activation energy. And so they need additional energy, usually as heat, to reach that transitional state. But you can't just heat up all chemical reactions to make them happen faster. Adding heat is not a useful way to speed reactions in cells because it can cause proteins to what's called denature. So if you remember the lecture about macromolecules and proteins kind of like are exposed to certain conditions, they will denature. Those bonds that hold it into that specific shape will break, and it might or might not be reversible. So adding too much heat is not really useful because you can also just denature the proteins in a living thing. Again, nothing's actually in a living thing. Nothing's in a closed system. None of these reactions are closed systems. And so, you can't just heat up everything. You're just going to denature everything itself. Heat is also impractical because it would speed up all the reactions in that cell, not just the ones that are needed. So you can't just rely on heat to speed up chemical reactions. So instead, organisms carry out catalysis, which is the process by which a catalyst selectively speeds up a reaction without itself being consumed. So an enzyme catalyzes a reaction by lowering the activation energy. And then it lowers it enough for the reaction to occur at moderate temperatures. An enzyme can't change the free energy required, but it speeds up a reaction that would eventually occur anyway. So here is an example. The black line is the course of a specific reaction without an enzyme. So this is the activation energy, EA, without any enzyme. This is kind of like the hump that you have to roll this ball over to then roll downhill. And then this red line is the course of this reaction with an enzyme. And so it lowers the activation energy. So it lowers like the barrier for this to happen, and then it can happen sooner in that moderate temperatures. So when enzymes recognize something, it usually is very specific. So the reactant in this chemical reaction that the enzyme's like helping speed up, the reactant that one enzyme acts on is called the enzyme's substrate. So the enzyme binds to its substrate and forms an enzyme-substrate complex. And so while they're bound together, the activity of the enzyme helps convert the substrate into the product of this chemical reaction. So here is a picture from the OpenStax textbook. So here's an enzyme, this grayish blob, and this is a substrate, kind of just generic. And so this is the one substrate that this enzyme binds to and recognizes. And so there's what's called an active site. That's where the enzyme, like the spot where it recognizes the substrate and where it binds. So when it binds, this is now an enzyme-substrate complex. And then it can help this reaction happen. And so now this reactant can be made into the products. So yeah, while bound, that activity of the enzyme converts that substrate into product. And so you'll see throughout your bio career that most enzyme names end in ACE. So just like how a lot of sugars end in OSE, a lot of enzyme names end in ACE. So in this case, sucrase is the enzyme that hydrolyzes sucrose into glucose and fructose. And so every enzyme catalyzes one specific reaction and can recognize its specific substrate even among closely related compounds. So sucrase only recognizes sucrose. It doesn't recognize other stuff that's like structurally similar, even if it's an isomer. It's very specific. The sucrase only recognizes its substrate, sucrose. And again, the active site of an enzyme is the region on the enzyme that binds to the substrate. It's usually like a pocket or a groove. So again, remember, even though we represent all kinds of things as just like blobs, that's not actually how they look. So an enzyme, this is probably even still simplified. So an enzyme is a protein. So remember, proteins are made of a bunch of amino acids put together and folded together multiple times. So this folds into like a very specific shape. And so the active site has a very specific shape that only recognizes that substrate. And so it'll often be in like a pocket or a groove. And so the fit between the active site and the substrate is responsible for how specific an enzyme is. It'll only work on this. When that substrate enters the active site, the enzyme changes shape slightly, kind of like tightening around the substrate, almost like a handshake, like when you shake hands with somebody. It's hard to do this with yourself. So you're shaking hands with somebody. They don't just fit together and then like that, you like squeeze each other's hands a little bit, right? So kind of like that. So when a substrate enters the active site or one hand goes with another hand, it kind of like holds on a little bit. So this is what's called an induced fit. And this results from those interactions between the chemical groups on the substrate and on the active site. And so the induced fit brings the chemical groups at the active site into positions that enhance catalysis of this reaction. So the substrate is typically held in the enzyme's active site by weak bonds, like hydrogen bonds. Usually it's reversible. And the conversion of substrate to product happens really fast, rapidly. And then the product is released from the active site. And again, enzymes don't get used up in the reaction, so once it lets go, it's ready to work again. So because enzymes emerge from reactions still in their original form, small amounts of enzyme can actually have huge impacts because it's going to keep working. It's not going to get used up in a reaction. It lets go and it's like ready for the next. So here is another example of another blob. So this is an enzyme, and then it happens to have two substrates in this example. So the two substrates fit in the active site. They're held in place by weak interactions, so they don't just float away. The active site lowers the activation energy of this reaction, and so that helps this process happen. The substrates are now converted into products, and then these products can just be like spit out or released. And then the enzyme is still around and available for new substrates. And so how do these enzymes lower the activation energy? So substrates can be oriented in certain ways to facilitate the reaction. Substrates can be stretched to make the bonds easier to break. the active site might provide a microenvironment that helps favor this reaction. And then the amino acids in the active site might even participate in the reaction. So it just depends on the enzyme, how it's actually lowering this activation energy. But it's doing something that kind of makes this reaction want to happen. So the rate of an enzyme catalyzed reaction can be sped up by increasing the substrate concentration. So say you have this reaction happening and you have enzymes in there, but you want it to happen even faster. So you can add more substrate, and then this happens to push, it to happen more. So the rate can be sped up by adding more substrate. Because as many enzymes are in there, they might not all be used, right? So like if you have a bunch of workers on staff, they might not all be actually being put to use. But if you add more substrate, then that gives more work for all the enzymes and the reaction happens faster. So when all enzyme molecules have their active sites engaged, then this enzyme is saturated. If the enzyme is saturated, that reaction rate can only be sped up by adding more enzyme because all the workers are busy. The only way you can have it happen even faster is just by adding more workers. And there are other things that affect reaction rate and enzyme activity. So enzyme activity can be affected by other general environmental factors. So not just whatever's participating in this, but stuff like temperature and pH can affect enzyme activity. It can also be affected by chemicals that specifically influence the enzyme. We'll break down each one of those things. So temperature. Each enzyme has an optimal temperature at which it'll catalyze its reaction at the maximum possible rate. So it's kind of like a Three Little Bears scenario that having just the right temperature will have that reaction happen like the maximum optimal rate. So up to that point in the optimal temperature, the reaction rate will increase with increasing temperature. But at a certain point, it's going to stop. So you can increase the temperature, and then up to this point, the reaction rate increases with the increasing temperature, but at a certain point, past the optimal temperature, the rate of reaction begins to drop. And that's because the enzymes will begin to denature beyond their optimum temperature. And that optimal temperature of an enzyme is going to depend on the environment that that organism and that enzyme typically is at. So for example, the optimal temperature for human enzymes, so any enzymes in our, in human cells and bodies, is going to be about 30 to 40 degrees Celsius because that's our human body temperature. So it makes sense that over time and natural selection, all of our enzymes would function optimally at our core body temperature. Our temperature is 37 degrees Celsius for body temperature. So yeah, all of our enzymes are going to be optimally functioning at our body temperature. But for other organisms that live in extreme environments, they're going to need higher temperatures or maybe even lower. So thermophilic bacteria are bacteria that live in super high temperatures, and so their optimal temperature can be higher because through natural selection, those enzymes have just come to be able to withstand those temperatures. So the optimal temperature for those are about 75 degrees Celsius, whatever environment that kind of bacteria typically lives in. So this is kind of comparing those two things. So the black line on the left is showing optimal temperature for a typical human enzyme. So if you increase the temperature, the rate of reaction will increase up to a certain point. the optimal temperature where you have the highest rate of reaction. And so in human enzymes, yeah, it's going to be 37 degrees. This red line to the right is going to show you a thermophilic bacteria's enzyme. So the temperature is going to be even higher. So as you increase, it'll go up. And this one up to 75 degrees Celsius. And then again, past that, the enzymes will denature. So it just depends on what the enzyme is and what organism it typically is in. So each enzyme has an optimal temperature, and each enzyme also has an optimal pH that is dependent on the environment in which it's typically active. So in this case, even in one human, we might be the same temperature, but different parts of our bodies have different pHs. So for example, the optimal pH for pepsin, which is a human stomach enzyme, is pretty low, really acidic, 2. Your stomach is very acidic. So this enzyme happens to be able to withstand a low pH like that. But the optimal pH for a different enzyme that's in your intestine, trypsin, is pH 8. So it just depends. It depends on the enzyme and where it typically is, and what natural selection has led it to be able to withstand. So here's that example in a picture form. So pepsin, the stomach enzyme, is going to have an optimal pH really low, really acidic, because it's going to be in the stomach. And then trypsin in the intestine is going to have an optimal pH at 8. So it just, again, depends on the enzyme and where it is, what it does. And so enzymes can do their job on their own, but it depends. So some enzymes might need cofactors. So cofactors are non-protein helpers that bind to the enzyme permanently or reversibly with the substrate. There are inorganic cofactors, and those include metal atoms like zinc, iron, and copper in ionic form. And there are also organic cofactors, but because they're organic, we call them coenzymes. So most vitamins either act as coenzymes or provide the raw materials needed to make them. So that's why you might see some of these as recommendations in your diet, because literally your body needs them to serve as cofactors for enzymes. And so there are certain chemicals that can selectively inhibit the action of specific enzymes. So many inhibitors will bind to the enzyme by weak interactions, resulting in reversible inhibition, and I'll show you some pictures on the next slide. So usually it's weak interactions. Again, remember, there are strong bonds between chemicals and weak bonds, and sometimes it's better to have weak bonds. So many inhibitors bind to enzymes by weak interactions, and then it's reversible. If an inhibitor binds with a strong bond, like covalent bonds, then the inhibition is usually irreversible, and now this enzyme can't do its job. So there are different ways that something inhibits an enzyme. So competitive inhibitors, this is just a term for something that inhibits something, but it might have an actual name. So here's just a generic enzyme and its substrate normally on the top in panel A. In panel B, there might be some kind of other molecule that serves as an inhibitor. And so competitive inhibitors closely resemble the substrate, and so it can bind to the active site. It kind of tricks its way in a little bit. And then that inhibitor blocks the substrate from entering into the active site. So now it's like competing, and it's like out-competing the normal substrate. So the inhibitor blocks the substrate from entering the active site, and the enzyme activity is reduced. So this particular enzyme has a competitive inhibitor, and so this enzyme can't do its job. There might be other of this specific enzyme floating around that hasn't been blocked by an inhibitor. So those might still work, but this one right now is blocked. So enzyme activity in total is reduced. And you can increase the reaction again by adding more substrate. So competitive inhibitors kind of compete for that active site. Non-competitive inhibitors bind somewhere else. So proteins, again, are super complex, and enzymes are really complex. And so they might have other sites. So in non-competitive inhibition, the inhibitors actually bind to a different part of the enzyme away from the active site. So it's not competing for the active site, it binds somewhere else, but then that causes a shape change in the enzyme and makes the active site less effective, and so now the enzyme activity is reduced. So non-competitive, it's not competing for the active site, it's binding somewhere else to inhibit. And so, for example, about how they are usually reversible. In cases where they're not reversible, it's bad. So toxins and poisons are often irreversible enzyme inhibitors. So for example, sarin gas was used in a chemical attack in Syria in 2017, killing and injuring hundreds of people. And sarin actually binds covalently, so again, a strong bond, to the active site of acetylcholinesterase. And this enzyme is important in the nervous system. So if it binds to the active site, it's a competitive inhibitor. And if it's binding covalently, it's not going to let go. It's a strong bond. And so that's how it disrupts the nervous system normal function and can kill and injure people. So other examples include pesticides and antibiotics. So again, sometimes having weak bonds and interactions is helpful. It's reversible. So how do we have all these different enzymes for all these different reactions? So enzymes are proteins, just like other ones, and they are coded by genes. So the genes go through the processes of transcription and translation to be made into proteins. And if there are changes in the gene, so mutations, this will lead to changes in the amino acid composition of the enzyme. So in a future lecture, we'll get into gene expression and how this happens. But yeah, changes in a gene, the recipe for a certain protein, end up leading to changes in the amino acid composition of the enzyme, and then altered amino acids, especially if it's at the active site, can result in new enzyme activity or altered substrate specificity. And if a mutation results in a new enzyme function that happens to be beneficial to the organism, then natural selection will favor that mutated allele. So for example, repeated mutation and selection on the beta-galactosidase enzyme in E. coli resulted in a change of the sugar substrate that this enzyme worked on. So ends in ACE, it's an enzyme. Beta-galactosidase maybe works on a certain sugar called beta-galactose. And so repeated mutation and natural selection can change what the enzyme's substrate is. And so this is a figure of that experiment that kind of showed this. So if this is the active site, they were able to pinpoint the 2 changed amino acids that were in the active site and resulted in these changes. And there were also some changed amino acids near the active site or on the surface, and so maybe these all work together to let it recognize a different substrate. Okay, and so how does this, again, this is all out of context. What? No, it all comes together. So again, we're talking about chemical reactions, and we're going to set the scene for metabolism. And so it's enzymes that regulate metabolic processes. Chemical chaos would happen if a cell's metabolic pathways were all happening at the same time. There has to be like control over all these different processes happening in a cell. So cells can regulate metabolic pathways different ways. You can, number one, switch on or off the genes that make enzymes, or you can also regulate the activity of the already existing enzymes. So it's kind of like back to the worker thing. You can hire less workers or more workers if you know there's going to be like extra need or less need at a certain time of the year or whatever. Or you can also regulate the activity of the existing workers. Like say there are already workers there and they're working like, maybe you incentivize them, give them more reason to like work better and be more efficient. So yeah, you can either switch on or off the genes that code for certain enzymes or regulate the activity of the existing enzymes. So there's this concept called allosteric regulation. It has nothing to do with aloe vera. So this occurs when there is some kind of regulatory molecule that binds to a protein at one site and affects the protein's functions at another site. So this type of regulation can either inhibit enzyme activity or stimulate enzyme activity. So this figure is kind of like a lot at once, but if we focus on the top side, let's say the top left. So this is an active form of this enzyme. So this enzyme actually has 4 subunits. So this whole enzyme has 4 subunits, 4 purple blobs. And each blob is in a subunit and has its own active site. So this is the active form of this enzyme. And then below in the bottom left is the inactive form. And now you can see there's a change and there's a non-functional active site. So this complex will switch between 2 shapes, the active form and the inactive form, based on what binds here. So an activating or inhibiting molecule can bind to a regulatory site. An activator stabilizes the active form of the enzyme, and an inhibitor stabilizes the inactive form of the enzyme. And these are, again, just kind of terms that talk about what it does. The actual activator can be a bunch of different things. There's not just one molecule called activator. And again, same for inhibitors. It can be different molecules depending on the enzyme. But in general, an activator stabilizes the active form of that enzyme. Inhibitor stabilizes the inactive form of that enzyme. Okay, so in cooperativity, no, let me go back. So this is going to show that. So an activator, shown in green, stabilizes the active form. And alternatively, there's an inhibitor that might stabilize the inactive form. And so now these active sites are not functional. So that's allosteric regulation. Again, when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. In this other type of allosteric regulation, it's called cooperativity. And so in this case, the substrate binding to one active site triggers a shape change in the enzyme that stabilizes the active form for all the other sites. So it's not like a different regulatory molecule, it's the substrate that is making this change. So this will amplify the response by having this enzyme more likely to act on additional substrate molecules. That is cooperativity. And so that is one focus of regulation. In feedback inhibition, the end product of a certain pathway will shut down the pathway. So if this is a pathway to make isoleucine, an amino acid, you have a bunch of different enzymes involved in this whole pathway with different intermediates in between. And so the easiest way to regulate this pathway and not make too much isoleucine is by having your end product feedback and inhibit the pathway. So to make isoleucine, you start out with this other substrate called threonine. And so this enzyme modifies it into this intermediate. Enzyme 2 modifies that into intermediate B, and then there's a whole pathway, right? And at the very end, there's the end product, isoleucine. And so that can go back, feedback, and this is the inhibitor for this allosterically regulated enzyme. And so now the active site is no longer available for that substrate, threonine, and then that stops the whole thing. The earlier you can stop a pathway from wasting energy, the better. So in feedback inhibition, the end product of the pathway feeds back and shuts down the whole pathway. And so this is really helpful. It helps prevent a cell from wasting chemical resources by making more product than is needed. So it's super helpful. And then lastly is where the enzymes are in a cell makes a difference. So localization enzymes. The compartmentalization of a cell, especially eukaryotic cells, right, that have compartments, helps bring order to these metabolic pathways. It literally separates different reactions. So in some cases, the enzymes for several steps in a metabolic pathway will form this huge multi-enzyme complex. Some enzymes have fixed locations and act as structural components of certain membranes. So what reactions are going to happen where depends on where the enzymes are. And so in eukaryotic cells, again, some enzymes will reside within specific organelles. So the enzymes that are important for making energy are going to be inside the mitochondria. And that's why the mitochondria is the powerhouse of the cell, because the enzymes that help these reactions happen are in there. So here's an example. Here's a cute little cartoon of the mitochondria, and then here's a real one. So inside is the mitochondrial matrix, and that has enzymes that are involved in the second stage of cell respiration. And then there are enzymes in here in the inner membrane that are important for the third stage of cell respiration. So yeah, different parts of organelles even are going to have different enzymes and have specific reactions happen. Okay, so that is everything. That's lecture 6, Introduction to Metabolism.